And thank you for inviting me to be a speaker. It's a pleasure to be here today. So one of the challenges for me in terms of speaking to a group like this is that I'm sure your background in science and health is pretty diverse, and so I'm going to keep things relatively basic. If you have more detail oriented questions because you have a background in this, by all means feel free to ask. I do have my chat box open in the corner, so I can keep an eye on that as I go. I've tried to keep this relatively informal, so we can approach it a little bit more like a discussion. But by all means, if you have questions, just go ahead and type them, and I can pause and answer them as we go. Okay. And so, in terms of what I want to cover today, is just a little bit of a background about vaccines as a whole. I think people know a little bit about them, but we'll go into a history a little bit, then we'll go into a little bit more detail in terms of how they work, um, and then we'll shift and a little bit talk about vaccines in the context of coronavirus, because I think what's in the news a lot is about these RNA vaccines and the new vaccine that's currently they're requesting to get approval, which is the Johnson and Johnson vaccine. That's actually a slightly different vaccine, so we'll talk about that as well. That's what's called the adenovirus vaccine. And then the other worry right now is what about all these new strains, and how does that impact what's been done and what's going on right now? So with that, I will start with a little bit of history. So the history of vaccines is actually quite old, but you have to keep in mind that a lot of this was based on observation, right? The idea behind the complexities of the immune response, even the understanding of what viruses are, didn't exist when vaccines actually first were developed. So a lot of the history tracks back to this guy in the upper left corner. This is Edward Jenner, and that he is basically credited for really putting a lot of observations kind of into practice. And so the idea simply here was that one of the major kind of plagues of that era was smallpox, right? And so smallpox was this disease caused by a virus that you know killed about 30% of the people that got infected, but many people could still survive it. And one of the things they noticed is that if you survived smallpox, you weren't going to get it again, right? And that's one of the keys here, and that ends up being kind of the basis behind vaccinations. And so the other major observation here was that dairymaids were for some reason protected from smallpox, and nobody really understood why at the time, until they made this observation that dairymaids were the women that were milking the cows, and they noticed that cows had a related disease called cowpox, right? But the key here is that cowpox, while they could infect humans, again, typically dairymaids because they were the ones in close contact with the cows, cowpox did not cause severe disease in humans, right? Clearly, humans could get infected, but it didn't have the debilitating pockmarks. It didn't kill people. And that he kind of pieced together that, well, maybe the infection from cowpox were actually protecting the dairymaids from smallpox, right? And it was that kind of correlation there that led him to actually test this hypothesis. So obviously, this sort of strategy would not fly in today's world with medical ethics and whatnot. But he basically infected a boy purposefully with cowpox because there was a dairymaid that had a lesion, and he basically took some material from that lesion and infected the boy. And then a couple months later, he tried to infect the boy with smallpox, right? And again, this would never fly in today's medical ethics era, but the little boy was immune. Okay. And so this was kind of that first demonstration that you could actually purposely infect someone with something, and that would lead to protection from something else. Okay, so his results were again published at the end of the 1700s. Um, and again, this was a big step forward kind of in the whole scheme of things, right? And so at the time, there was some skepticism, right? And this is actually an old piece of art from the early 1800s that you can kind of see depicted him vaccinating these people, and you can see they were slowly turning into cows. And so there was a lot of kind of skepticism around this idea that you could vaccinate someone. But ultimately, he coined this term vaccination, vacca from cow, right? Vaccinia was the virus. Now we know that causes cowpox. So, as time progressed, it it was hard to argue with the results, and that towards the late 1800s, this became fairly common practice. There would be scientists and physicians basically trying to go around collecting cowpox samples and using them to inoculate people. And again, here the hope was that they would ultimately be protected from smallpox, right? So why does this work? Well, we have to kind of take a step back and say, okay, why, do um, we get sick in general? And when we get sick, what actually happens? So in all of us, we have kind of an immune system. And so when we talk about the immune system as a whole, it's an incredibly complex thing. And I don't want to get too lost in the weeds here. But the thing to keep in mind is that the immune system is actually made up of a lot of different cells, right? And that when we look at the immune system as a whole, we usually break it down into kind of two groups. One is what we call an innate immune system, and then one is what we call an adaptive immune system. Now, humans and complex vertebrates have both an innate and an adaptive immune system. Simple organisms like invertebrates, insects, plants, they only have innate immune systems. So, kind of the adaptive part of the immune system is just characteristic of more complex or more evolved organisms. But it's that adaptive immune system that actually explains why vaccines work. Okay, so again, the idea simply comes down to the concept that our immune system actually can learn, right? And it learns and it has what we call a memory, right? So what does that really mean? It means that when we look in the context of the adaptive immune system, it's named because it can adapt to the different things we're exposed to. So when we're exposed to something, our immune cells get activated, and that during this activation process, they're going to try to get rid of that pathogen. But after they clear that pathogen, those immune cells are left behind at a kind of heightened state of awareness, also in greater abundance. So if we see that same thing again, we won't get sick, right? And that really comes down to why vaccines work. The hope is that we expose our immune system to something that doesn't necessarily make us sick, but it still elicits an immune response. But if we see something that's very similar to what we saw the first time, that the memory, if it's close enough, will actually protect us from the thing we actually worry about. So in this example where cowpox doesn't cause significant disease in humans, smallpox does, but cowpox is similar enough to smallpox that if we see cowpox, build an immune response, that immune response will actually protect us from smallpox. The key is that those two things have to be close in terms of what they are. So for example, if I get the flu, the virus that causes the flu is very, very different than, let's say, the virus that causes rabies, right? They have to be related enough so that your immune system sees enough similarity between the two things that concept works, right? And the same things, historically we've seen, I remember when I was a kid, chickenpox. We didn't have a vaccine for chickenpox, but the idea was that if you got chickenpox as a kid, it really wasn't that big a deal. But the key is if you got chickenpox as a kid, you wouldn't get it later on in life. And the idea was if you got chickenpox later on in life and never had it as a kid, that was going to cause you more problems. Okay, so the key point here is that it's the adaptive immune response that protects us in terms of long term. And when we talk about adaptive immune systems, the cells that are the primary mediator of the adaptive immune response, what we call B cells here, and what are called T cells. T cells fall into two categories, but I don't want to get into too much of the detail there. the major thing to think about are B cells make antibodies. So when you hear about, oh, we have all these antibodies, blah, 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 B cells are the cell types in your body that's making all those antibodies. Okay. Now T cells have a little bit more complex role. T cells actually circulate around the body and they look for virally infected cells and they will kill virally infected cells. And that's again their primary job. And so the B cells and the T cells are really the reason vaccinations work in us because these cells have, again, a memory. They remember what they've seen in the past, and therefore they can protect us if we see that thing again, either the same thing or something very, very similar to that thing. Okay. Okay, so here's kind of an example of what's going on here where this is kind of a measure of antibodies, let's say. This is abundance of a certain antibody in your body. And so if you see a pathogen, here we're just calling it an antigen, right? This is something that our immune system might key in on. You can see that after a couple days of exposure, we start responding. And that's what's shown kind of here in the blue curve. You guys can see my arrow as I point to the slide. Right. So we can see the curve start going up and simply saying, okay, we're going to start making antibodies if we see some sort of pathogen, right? And that's, again, a good thing. After we clear that pathogen, you can see that the antibody levels will go down. But the key is they don't go down to where they started. They actually go down to kind of a basal state that is higher than if you had never seen it. And the trick here is if you see that same antigen again in blue, you get a much faster response. You can see that time in days is on the axis down here. You get a faster response and it's even a much, much better response. Okay, so the hope is that by exposure to something the first time, you get your response. If you see it a second time, the response is faster and better. And the reality is they only show 2 here, but you could actually see it a third time and get even better, right? So this phenomena kind of explains why vaccines work to begin with. This could be your vaccination. This could be the exposure to the pathogen in real life. But this could also be your first shot. This could be your second shot because this is actually going to be left behind also at a much higher level. So the more times you see or exposed to a pathogen, the better your immune response gets. Okay, so this is an example of antibodies, but this is also true for the number of T cells that, remember, T cells are going around trying to kill virally infected cells throughout your body. Okay, so we go from a relatively slow response from a few days, and during this time, the virus is, typically replicating very rapidly. But if we see it again, you can see that it happens much, much shorter. And so the response time is so much faster that we can actually typically clear the virus before they really start causing any problems. Okay, so here again is an example where cowpox and smallpox are very, very similar viruses. If we see cowpox, we're going to make antibodies against cowpox, and that's kind of shown here in these little Y-shaped molecules are meant to depict antibodies. We have a blue one and a red one and a green one. You can see that smallpox is similar, but it is different. But it's similar enough that some of these antibodies cross-react, meaning in an antibody that was designed against cowpox by your immune system can also still recognize smallpox. And that's why the similarity matters, right? If you say, again, let's say one of these was the flu virus and one of these was rabies, they're so different that antibodies generated from one wouldn't be able to recognize the other. Okay, so again, the key to vaccination is our adaptive immune response. The hallmarks of our adaptive immune response are B cells because they make these antibodies. These antibodies stick around. There are some antibodies that you made decades ago that are still being made now, right? And so that keeps us protected. And then there are also T cells. T cells get less kind of press in the news about protective responses, but T cells are very important in vaccination as well. Okay, so again, antibodies can stay around for long periods of time even after the virus has been cleared. And that's also one of the ways we can detect prior exposure. For example, let's say you remember getting sick back in, March, right? And I'm like, well, coronavirus wasn't really in the news back then. And, you got a little sick, but then you felt better. A couple months later, we can actually go draw some blood, and we can see, do you have antibodies towards the coronavirus? If you do, that meant that you were exposed at some point in time. It doesn't mean you have the virus now, but the presence of antibody indicates that you had been exposed at some point, right? And that's why, again, in the news, people are talking about, oh, if I'm getting a coronavirus test, what are we testing for? Are we testing for the virus? There were some tests that were actually looking for antibodies in the blood. Those aren't commonly used anymore for a variety of reasons. But there were all sorts of different tests simply to say who had been exposed, who had ongoing infections, and we can talk about the tests if you have questions about the tests as well. Okay, so now let's get back to another kind of question. What is a virus, right? Why do viruses cause such real problems in us? Well, the key to viruses is they're incredibly diverse in their genetic makeup, their size, their shape, and also their pathology that they cause, right? Some viruses don't cause us any problems whatsoever. Other viruses are very, very deadly and kill us very easily. So why is that? Again, it depends on the nature of the virus itself, but all viruses have kind of one goal. And what I kind of, convey is that the goal is simply for those viruses to replicate. We don't think of viruses as being alive, meaning that if I had, a tube that contained viral particles and I knew exactly how many viral particles were in that tube, that number is not going to change. They can't make more of themselves unless they have a host. And when I say a host, basically a cell, right? Viruses can have very, very few genes, meaning that they're the simplest viruses out there that only have 4, 5, 6 genes. And then there are some viruses that are more complex that have literally hundreds and hundreds of genes. For a comparison, humans, we have 25,000 genes or so, right? So viruses can be very, very simple because they are not technically alive. I always, again, kind of explain it by they're kind of like a molecular parasite. They parasitize a cell, they utilize the cell to ultimately make more of themselves. Okay, so ultimately, again, they infect a cell. You can see again in this diagram, here's the viral particle itself. Here's the picture of a cell. The virus goes into the cell, and once the virus is in the cell, it releases its genome into the cell. And during this process, the genome basically tells the cell how to make more viral particles. Okay, so ultimately, the virus kind of commandeers the cell to make more viral particles, and more viral particles are ultimately going to be released. Now, our immune system can see the viral particles themselves. Our immune system can see the infected cell that's trying to make viral particles. And so, I mentioned earlier T cells. T cells can actually identify virally infected cells and try to kill them in order to limit the production of viruses. Okay. So what is currently used as a vaccine? So when I say currently, I meant before coronavirus, right? There were a couple categories that vaccines would fall into, and it again depended on what the pathogen was. Now, one of the best types of vaccines prior to the coronavirus pandemic were what we call live attenuated viruses. When I say live, I don't mean alive like a living organism. I mean live as in infection competent, right? Meaning that these viruses could infect living cells. Now, the word attenuated here simply means weakened, right? Here, these were the viruses that could somehow be weakened, and then we could use them as a vaccine, right? So, for example, people could take something like a polio vaccine or a polio virus, sorry, and manipulate it to try to weaken it so that it either would grow really slowly or not cause the disease, but it could still maybe infect humans at a very slow rate. So our immune system would see it and we would develop an immune response. So this live attenuated virus vaccine, these were some of the best vaccines out there. The biggest challenge was it takes a long time to develop these because we have to understand why the virus makes us sick. And that's not always an easy proposition, right? Again, why some viruses can make us really sick and others don't can be very, very complex. But the fact that they are trying to replicate means that the immune system is activated really, really robustly, right? So here's kind of the cartoon diagram. Let's say we isolated a pathogenic virus, and kind of in this green, red, yellow, that's kind of a cartoon diagram of the genome of the virus. So let's say we could figure out which part of the genome causes virulence or causes the disease to manifest. If we simply mutated this region or simply got rid of that region, then we would have a virus that could still infect, but it simply wouldn't cause disease. And that would make a great vaccine. Okay. But again, the challenge with this approach is we have to understand a lot about the virus. We have to understand how it replicates, why it causes disease. And that typically takes years and years of research to really figure out why viruses cause that problem. Okay. The other way to do it is collect this virus and we can simply inactivate it. This can either be done through, boiling it. We could add different chemicals to create this dead, and dead simply means a non-replication competent virus, right? So, and dead inactivated virus. So, for example, your seasonal flu vaccine is typically a dead inactivated virus. If you're getting a shot in the arm for a seasonal flu, that's usually a dead inactivated virus, right? If you've ever taken the inhaled nasal flu vaccine, that's actually a live attenuated vaccine, right? So there are some pros and cons to both, but that kind of gives you an idea of the technology before. And then one of the last things here is a pure protein from the virus. If we could simply purify one of the proteins, we can use that as a vaccine. It's probably more commonly done for bacterial pathogens, but it can also be done for viral pathogens. Now, the challenge with these things, especially the first two, is that we have to know a lot about the virus and we have to be able to grow massive amounts of this virus, right? How we make flu vaccines actually use fertilized chicken eggs. So the flu vaccine is grown in huge, huge quantities in fertilized chicken eggs. Now, that's why they always ask you, do you have an egg allergy? If you're trying to get your flu vaccine, because it's actually made from chicken eggs. And so the ability to grow huge amounts of virus, that's quite challenging. And so that's why there's kind of been a push to try new uh, technologies for developing vaccines. And that kind of takes us to what has been in the news now. There are what are called mRNA vaccines, and this is the Pfizer and the Moderna vaccine. And then more recently, there's this adenovirus vaccine. And this is the AstraZeneca vaccine that's approved in the UK and placed in Europe, but not in the US. And then the Johnson & Johnson vaccine, which I think requested approval a couple days ago. And so these are in the works right now. These two have actually been approved. Okay, so let's go through really quickly what these are versus what the old vaccines were. Now, let's talk about what RNA is really. I think most people have heard the term DNA and they're comfortable with the idea of DNA. DNA, again, is kind of the blueprint of us as a living organism. All independently living organisms have DNA. The key here is when DNA or when cells divide, DNA has to replicate. So here's a cell with some DNA in it. Before that cell divides, the DNA has to be copied or replicated, right? That means that when the cell divides into two new cells, each new cell has the same copy of DNA as from what we call the mother cell, right? So that means that DNA is constantly replicated, and that you can imagine if this is a fertilized egg, right? And as that one cell develops, we're going to develop into a large multicellular organism. So let's take this mouse, for example, right? This mouse originated as a single fertilization event, right? An egg and a sperm led to the production of this mouse. And if this is true, every cell should have the same DNA. But if I took cells out of this mouse, let's say I looked at the immune cell, let's say I looked at a neuron or an epithelial cell, so something from the cheek, you can see that all of these cells came from the same mouse. So in theory, they have the same DNA, right? But if they have the same DNA, why do they look different, right? Why do they do different things? Again, we're always taught that the DNA is the blueprint of everything, and that is true. But a neuron does something very, very different than what an immune cell does. But they have the exact same DNA, so how can that be? The answer to that question turns out to be it's about RNA, right? So what is RNA in the context of all of this? You can imagine RNA is kind of it's which parts of the DNA are actually going to be used. Just because the cell has the entire genome in it doesn't mean that it utilizes the entire genome, right? So what that means is that here's our DNA. Every time this DNA gets used, we make RNA from the DNA. this RNA can then be used to make proteins, right? And so every cell has the same DNA, but not every cell utilizes RNA from all of those genes. So genes are embedded in our DNA. For example, like I said, we have about 25,000 genes in our genome. A given cell, like a liver cell, might actually only be using 20,000 of those genes. Not every gene is used in every cell. If we go back to the analogy with the blueprints, I could say, okay, DNA is our blueprint. But if I'm building a house with this blueprint and I hire, let's say, an electrician, the electrician looks at the blueprint, but it doesn't need every page of that blueprint. It only cares about where the wiring is. It doesn't care so much about where the sewer lines are. It doesn't really care about, where the roof is going to lie, right? If I hire obviously a plumber, they're going to care more about the pipes and the water and whatnot. They don't really care about the wiring. So that's kind of the analogous situation here where the DNA is like the blueprint. That's all of the information, but not all of that information is going to be used by every cell all the time. Okay, so one of the ways we can kind of make this a little bit easier is simply we can use RNA and we can directly administer that to the cells that we want, right? So instead of trying to get cells to take up DNA, we can simply get cells to take up RNA. RNA is basically taking out that step, right? So ultimately, what's happening here is here's an image of the coronavirus, and here's the genome of the coronavirus. The coronavirus genome is not particularly complex. It's kind of big, but not, not crazy. We know one of the genes that's shown here in green called the S gene. The S gene codes for the S protein, and S stands for spike. I think you guys might have heard about spike protein in the news. That is this green protein that's kind of on the outside of the virus. And the reason this protein is important is because it's on the outside of the virus, and that is what our immune system sees, right? Our immune system simply looks at this thing and says, okay, I see these big green things. Let's mount an immune response toward these big green things. And so what the scientists basically did was they took simply the gene that encodes for this green thing, right? And they, add a few other things into it, but they directly inject just that green gene, right? So by taking just the RNA of this green protein, they're not worried about, it's not going to make you sick. It's not going to create a full viral particle because the full viral particle needs the entire genome. They're just taking the one thing that the immune system sees really, really readily. And that ultimately your cells start making this gene or this protein, and your immune system sees it. It thinks it's actually a virus, but the reality is it's only a part of that virus that can't make you sick, but your immune system still sees it as if it were an infected cell. Okay. So some questions in the chat. How long will the vaccines be effective? And that's a good question. I'll talk about that a little bit at the end. And I'll talk about that variant as well. Yeah, so we'll talk also about some of the worries at the end. Let me get through a little bit of kind of the details of the vaccines, and we'll spend some time talking about kind of what people are worried about, and then the long-term protection. Okay, so after all these vaccines are developed, the question is, how do we test if they actually work? Now, in this day and age, we can't do what Edward Jenner did. We can't vaccinate people and purposely try to infect them. In the US, it's not considered ethical to do what's called a challenge trial. A challenge trial would be to vaccinate someone and then challenge them with a pathogen. There are too many complications that if your vaccine doesn't work, you don't want to purposefully make someone sick. So how this works is that they enroll a ton of people, right? So in this case, Moderna enrolled 30,000 people, Pfizer enrolled 40,000 plus people, and that they simply get these volunteers. Half of these people get the vaccine, right? The other half of these people get a placebo, right? And the placebo is just kind of a fake vaccine that doesn't actually do anything, right? And then they wait. So you give your cohort, some get vaccine, some get placebo, and then you just wait. And then after they waited a while, they said, okay, of my original 30,000, how many got coronavirus? And it turns out 196 of them got coronavirus out of that 30,000. But then the question is, if you look at the 196 people, how many were from the group that got the vaccine. And so you can see here that 185 of them came from the placebo group, and only 11 came from the vaccinated group. Again, these people don't know if they got the vaccine or the placebo, so that doesn't change their behaviors or anything like that. And what it tells you is that the vast majority of coronavirus cases were simply from people that didn't get the real vaccine. And if you got the real vaccine, you were pretty well protected. And so Pfizer got the same sort of, data where they had a larger group. They had 170 cases of coronavirus. 162 came from the placebo group, and only 8 came from the vaccinated group. So that's the kind of data that these drug companies are trying to gather to say, does this vaccine work? You simply give a group the vaccine, give a parallel group a fake vaccine or a placebo, and then you let them back into the population and see who gets sick. And that it's clear that the vast majority of people that got sick were the people that got the placebo, not the actual vaccine. Okay. Why do some people get sick after this? Let me go up a little bit. Okay, so one question that does come up a lot is that if you inject RNA to someone, does that affect their DNA? And the answer is no. And that is actually one of the reasons these companies went after RNA-based vaccines. There are DNA-based vaccines in research studies, nothing in human trials or anything. RNA is much safer than DNA, because if you put DNA into the cell, there's a chance that that DNA could integrate into that cell's own DNA. If you put RNA into a cell, RNA cannot incorporate into the DNA of the cell. And RNA is not very long lived in a cell. If you put RNA in a cell, it doesn't typically stick around for, more than, depending on the system, more than a few hours to a few days. Okay, so RNA is a much, much safer way to do it than using DNA, because DNA always has some risk involved in integrating into a normal genome. Why do some people get sick after the second shot? You mean sick from the actual coronavirus infection, or feeling sick from the vaccine shot itself? Well, I can answer it both ways. So some people get sick from the second shot simply because the immune response hadn't kicked in enough yet. So keep in mind that from the time you get your second shot, there still takes a few days for the immune response to reach its peak protection. And so a lot of this is still kind of in the timing of things, right? If you get your second shot, it's not that right after your second shot, you're protected. You still typically need to wait a few more days before you get full protection. But the other thing here is that just because you have 2 shots and you are protected, remember, this is still 95%. There's still 5% of the people out there that can still get sick, right? We know that there were 11 people out of the 15,000 that still got coronavirus. So this is not 100% protection, and it's not, it's obviously a lot better than if you weren't vaccinated at all, but it is not 100% effective. 95 is pretty good though, but that is one of the reasons why some people are still getting infected after the vaccine. And we'll talk about the variants as well, because the vaccines, while they do work for at least the current variants, there's some evidence that they're not as good. And we can talk about that a little bit. What is your response to people who think scientists are manipulating our DNA? I think there's kind of a big picture, question to that. What would they be trying to do, right? What would their purpose in doing that really be, right? And the reality is, manipulating our DNA is not that easy. It's easy to mess up our DNA. It's not that easy to change it to something we really, really want, right? So, we can damage DNA really easy. That's through chemical carcinogens, that's through ultraviolet light, that's through radiation, but it's very, very hard to change a DNA from something to something else in a purposeful way. So, do I worry that the government is doing that? Me personally, no. But I also, I kind of understand what the limitations are on manipulating DNA. It's not as easy as kind of they make it out in in the news. While things like CRISPR and genome editing do exist, it is still really in its infancy, and it's not something that um, in this format is something I would worry about. ADE in vaccines is a very good question. ADE is an antibody-dependent enhancement. Let me hold on to that one in a little bit, because that's a good question. Do we know how to treat people infected by the COVID-19 virus? You know, we have some strategies to treat it. There are, so let me talk about one strategy here real quick, is that, you know, I'm talking about vaccines in order to develop antibodies, and that those antibodies can protect us. One of the treatments was simply to administer antibodies directly, right? If we could actually purify large amounts of antibody and inject antibodies into people, then that would save all this stuff about vaccines. The problem with that approach is that the antibodies don't last forever. Antibodies only last in the body for, again, depending on what type, you know, maybe 100 days or so. Um, but the problem is, if you put antibodies into something, you can't make more of them without injecting more into a person. Okay, so you know that antibody therapy. I think many of you guys might have heard Regeneron. That was in the news a lot. I think the former president took it. Regeneron is basically antibodies that were designed in a lab, injected into a person, and that was to try to, you know, mimic the effects of a vaccine, because we didn't have vaccines at the time. How long after the second shot until you have max effectiveness? You know, it's the sort of thing where the clinical trials were done in a certain way that, depending on if you got Moderna or Pfizer, the recommendation was kind of after the first shot, you would have to wait either 3 weeks or 4 weeks, again, depending on which one you got, and then you would take the second shot. Typically, from that second shot, you would need at least a week after that. But you know, it's kind of a moving target in terms of some people will have max effectiveness, you know, a couple days before somebody else. So there's a little bit of variability just in the human population. But I would guess at least probably like a week after that second shot. Um, Efficacy to flu vaccines are much less effective. Flu vaccines, depending on the year, are closer to the 60 or so percent effectiveness. And that's for a complicated reason, because the flu vaccine typically changes from year to year depending on what flus are actually circulating. Now, as you've seen in the current coronavirus right now, it takes quite a bit of time that once we decide on a vaccine, we still have to make it in large enough abundance to distribute across the nation, right? That's kind of the limitation right now. So the flu vaccine, we've had enough experience with the flu vaccine that we know flu season is coming. Flu season is very seasonal. It typically starts towards the end of fall through winter, and then it starts to taper off kind of as things warm up in the spring. But since we have to vaccinate for the flu before the flu season really starts, scientists are actually guessing at which strains are going to be the predominant strains um, before flu season actually begins. So there's a lot of surveillance, meaning a lot of scientists around the world are looking to see which strains are out there. And when you take a flu vaccine shot, it's usually a mixture of 3 or 4 strains. It's their best guess as to which ones are going to be out there. Sometimes those guesses are right. Sometimes those guesses aren't 100% right. And that's why the flu vaccine is a little bit more variable, simply because a lot of it is actually based on a little bit of a guess, and simply just the way they're designed. They're not the most effective vaccine. Remember I said they were kind of these dead inactivated viruses, which aren't always as effective as other types of vaccines. Measles vaccine is very, very effective. And partly, and that's one of the reasons you know, we don't have measles outbreaks, assuming people are still getting vaccinated and whatnot. I don't remember the actual number, but it's very, very high. I want to say well into the 90%. I think more people having reaction to the second than the first. So when you say reaction, one of the issues is when you get an infection or even a vaccine, sometimes you get sore, right? Or you get a little bit of redness and a little bit of swelling. that's actually a sign that your immune response is working. And so that in some ways is actually a positive thing because your immune system usually kind of makes you feel crappy in order to allow your body to recover, right? And so that's actually when you get sick and you don't feel well, it's usually your immune response working because your immune system is trying to limit your activity, trying to make sure that you are resting enough to recover from these sorts of things. Okay. Let me go through a few more slides here because I don't want to get too sidetracked with the questions. Let me talk about the AstraZeneca and the Johnson & Johnson vaccine real quick, because this is also a novel idea in terms of human use as a vaccine. So this is the idea where we take a harmless virus. The type of virus here is used in adenovirus. So this is a harmless virus that doesn't really make us sick. But what we do is we take that spike gene from the coronavirus and we put it into the adenovirus. So we're simply taking one of the genes that the immune system likes to key on, but we put it in a virus that doesn't cause any disease at all. And so this type of virus ultimately doesn't cause disease, but the immune system still sees that one gene from the coronavirus that we put into this harmless virus. And because of that, it mounts an immune response to the harmless virus and to that one gene that we put into that virus. So that's the AstraZeneca version, the Johnson & Johnson. Now the key here is that it's one shot, right? Its storage is more convenient, it just has to be refrigerated, it doesn't have to be in these ultra cold freezers and whatnot. But if you look at the data, it's not as protective, right? The AstraZeneca, they argue, is about 70%. The Johnson & Johnson one's a little bit even more complicated because there were differences in different parts of the country. And this alludes to the variant questions, right? We know now that the South African strain is a variant and that might explain why the vaccine had lower efficacy in South Africa compared to in the US, where in the US we weren't exposed, at least during the time when this trial was going on, to some of these other variants, right? So there is some differences regionally. That could be due to the different strains out there. Could also be due to different genetic pools, right? The different genomes in different parts of the world. But you can see it's a similar idea where there are a large number of participants. They have half placebo, half vaccine, and then they see, okay, who gets sick. Okay, so one other thing I do want to mention here, it seems like, oh, it doesn't work as well, but the key here is a balance. Anytime a company does a clinical trial in this country, they have to tell the government what they're going to look at as their endpoint. They're going to say what they're going to call a successful thing even before they start. They chose to say, we want to do this with one shot, right? Now, if they had written their clinical trial to say, oh, we're going to do it with two shots, oh, I guarantee you that they will get much higher than 70%, right? The same would go for Pfizer and Moderna. If they had said, oh, we're going to do our clinical trial with only one shot, the data would probably show that it still works with one shot, just not as well, right? So part of this is that you can't compare this to the Moderna Pfizer numbers as well, simply because it's like comparing apples and oranges. If they had gone the two-shot route, this would be much, much higher. They simply were saying, well, we think one shot has an advantage. We're willing to take a decrease in its effectiveness simply by making this one shot. Okay. And then the last thing, will the vaccine still work against new variants? This is one of my, so the students I teach nowadays are, 18-year-olds, so they're born in the 2000s, which scares me a little bit. They don't know what a one-eyed, one-horn flying purple people eater is, but this is my kind of analogy, is that when your immune system sees a virus, different immune cells see different parts of the virus. So for example, if these circles here are immune cells, this one might be looking for, oh, things with one eye. This thing might be looking for things with one horn. This thing might be looking for one that can fly. This one looks for things that can eat people, right? So it's a collection of different immune cells that all key in on a pathogen. So the question is, if we start having variants, will the vaccine still work? And the answer is yes, to a point, right? If this guy turned red, well, if it still had one eye, sure, some immune cells could see it, right? But the one that looks for purple, wherever he is, wouldn't be able to see that one anymore. So it's, again, a collective phenomena where if more and more changes start to occur, then the vaccines will become less and less effective. Currently, the South African strain is the biggest worry, simply because while tests in labs have shown that the vaccine still does work, it doesn't work as well. So that, again, is kind of the big worry right now. And then, the questions about, you know, what happens long term, that's still the big one. So kind of let me summarize before I head back to the questions here. These are new vaccine strategies. The mRNA and the adenovirus vaccines are um, new to human use. They've actually been around for easily more than a decade in concept, in animal studies and whatnot. So they're not new in that regard, but they are new for use in humans. Some of them have actually been used in the veterinary world before humans. So, we do have some indication that they do work, right? And they're safe and effective, especially if we look at, so far all of the vaccine trials, they're monitored for safety and efficacy and whatnot. The other advantages to the new platform is it doesn't require large production of the original pathogen. Remember when I said if we're going to either attenuate a virus or if we're going to inactivate a virus, we have to be able to grow large amounts of the virus. To generate all of these vaccines, we don't have to grow coronavirus at all, right? We simply make the RNA, which can be done in a lab, or we can grow up large amounts of this harmless virus. This harmless virus has been studied for decades. We know a lot about it. We can grow it really, really easily. And so those are some of the benefits to these new vaccine strategies. We don't have to spend months to years to figure out how to attenuate a virus. This is a much, much quicker way to generate vaccines. The trade-off here is we don't know how long protection will last. And this is, again, a big unknown. Current clinical trial participants are going to be monitored for 2 years, right, to see that. But, it's been approved for emergency use because it's clear that there is a benefit, and that's why there still is a little bit, should we be worried about safety? You should always be worried about safety. But the question is that worry more than, the consequences of getting coronavirus? And that's really what it comes down to, right? So with that, let me go back to the questions here. Yeah, so people are mapping the coronavirus variants. There are huge databases that are publicly available. So the question is, yeah, are we mapping all of the variants? There's a lot of work being done on mapping the variants because since most of these vaccines are against that spike protein, that is where they're really worried right now. A lot of the new variants have changes in that spike protein. Um, you know, the key to slowing down the generation of variants is to slowing down infection as a whole. Again, if I had a tube filled with viruses, they cannot mutate sitting still in a tube. They can only mutate when they are actively infecting cells. So if we slow down infection rates, if we decrease the number of people that actually get infected, we will slow down the creation of these new variants. The evolution will slow down simply because that evolution is driven by the virus replicating itself. Work on this technology has been going on for a while. So you're right. So we first encountered coronavirus in terms of a deadly pathogen in the SARS in early 2000. So coronaviruses in humans had actually first identified in the 60s. Mid 60s, there were a couple coronavirus strains that were identified, but they didn't cause any real human disease. They caused cold-like symptoms. They didn't really worry anyone. And that's why when the SARS outbreak occurred, the major issue there was that it was fairly deadly, right? Now, the difference between the original SARS in the early 2000s compared to the coronavirus now is that this coronavirus is much, much more contagious. The first SARS was more deadly, but much less contagious. A lot of work went into developing new kind of strategies, studying the virus. So scientists learned a lot about the virus, but the actual issue with developing a vaccine to SARS was that it came and it went, right? Remember that the way to test if a vaccine works is by vaccinating a huge number of people and then waiting to see if they get sick or not. That wasn't able to be done with SARS because not enough people actually got it, right? So you can't vaccinate a bunch of people and see who got sick because SARS ultimately came and it went. So yes, a lot of research was done in understanding the SARS virus, understanding how it replicated, understanding how it caused disease, but it was a little bit harder to do vaccine studies. There were a lot of vaccine studies done in animals and mice, But again, it kind of went away before a lot of the vaccine technology really got up and going. Think back to like 2016. Remember the Zika virus, right? The same thing happened kind of there, where it was in the news a lot, but then as people started to develop the vaccines, it kind of went away. And so part of the ability to develop good vaccines means that the infections still need to be fairly ongoing to really do a good clinical trial. We could always test it in animals, but nobody is going to, the FDA would never allow a vaccine that had only been done in animals to go into humans in large numbers. You always start with a phase 1 trial, which is a safety trial. You do a couple, maybe a few dozen people. Then you do a phase 2 trial where you increase. It's always about safety at that point. And then the phase 3 trial, these are these large, 30,000, 40,000 people. And that's when you actually test to see if it works. Okay. What is the difference between Moderna and Pfizer? So part of that is the differences aren't always made public because a lot of the vaccine formulations are proprietary, meaning that the different companies have different, ways that they make it, and therefore they might not want to share that information. That presumably is why one needs to be stored in ultra cold. The other one can actually just be stored in a freezer. Is partly due to their formulation. The mRNA part is probably very, very similar. People have asked me in the past, if you got a Moderna vaccine the first shot and then could you get a Pfizer one the second one? The answer is I wouldn't do it simply because that hadn't been tested. In theory, would it work? In theory, it probably would be okay. But since that's not how it was tested, I would not do that, right? You're only as good as what you've actually been tested for, right? So if you got a Moderna shot the first time, get a Moderna shot the second time. Same thing. If you got the Pfizer shot the first time, get a Pfizer shot the second time. The difference is, does one have more side effects than the other? I haven't heard about. Both of them had actually much fewer side effects compared to the AstraZeneca trials. The AstraZeneca trials were a little bit kind of more all over the place. I don't know if you heard about some of the controversies about the AstraZeneca trial. There was some mistakes made in formulation and whatnot. But in general, the Pfizer and Moderna showed very similar safety profiles because they're very similar in their strategies. Thought breathing your own toxins that they may go into lung. So I guess the question is, what toxins are you worried about? At least for coronavirus, what we know now is that the spread is primarily based on aerosol droplets. So while it's still a good idea to always wash your hands and keep your surfaces clean, that's not the primary route. And so even though there was a lot of emphasis placed on disinfecting every surface in your house and whatnot, it's much more about being in a room next to someone who is infected, right? And knowing that, the easiest way to slow the spread of aerosol droplets is simply by wearing a mask. And we've seen the effectiveness of a mask in certain countries where the mask wearing is much more heavily enforced, much more mandatory. And I don't know if many of you guys have traveled to Asian countries, even well before the pandemic. Wearing a mask is fairly common in a lot of places. And so by minimizing aerosol droplets into the air, we know that ventilation plays a really, really big role. We know that, again, wearing a mask, because it minimizes aerosol droplets, plays a huge role in that. In terms of, does wearing a mask make you more sick? I don't know what toxins would be present in that context. Obviously, if you had some chronic breathing disease, wearing a mask that would restrict your breathing could potentially be a worry, but that would be, a fraction of a percent of a population here. In general, the vast majority of, people would not have any health consequences due to wearing a mask. So is the virus mutating? Yeah, the virus is mutating, and that's where these variants come from. Every time that virus replicates itself, there's a chance that it makes a mistake. So if we let me see if I can go back to, oops, let me go back. So if we look at this picture here, you can see that one of the things that happens is when the virus infects, the genome goes in, the genome actually replicates itself. When it makes more copies of itself, there's a chance that mistakes are made, and that's where mutations occur. So if we prevent viruses from infecting cells, then the virus has no chance to make more of themselves, and therefore it won't create mistakes. And that's what I meant, that viruses in a tube, they're not changing. Viruses can only change when they're actually infecting a cell. Okay, so that's why by preventing infections and decreasing the rates, we will slow the creation of these mutants or these new variants. Against the shots. I don't know what the question there was. When do I think the pandemic will no longer be a pandemic? That's a good question. You know, it's hard for me to know. I mean, vaccine distribution has been pretty slow. You know, me in terms of we're so at least at the university here, we're planning our fall schedule right now, and they're telling us that we will have some classes in person, but a lot of our classes are still not going to be in person. And so we're kind of planning for some mandatory classes, you know, like lab classes, art classes that have to be hands-on. Those will probably be in person in the fall. But, you know, I teach the sophomore biology class with 160 students in a lecture hall. That's not happening in the fall. So when this whole ends is still not really clear. I don't really even have a best guess for those sorts of, you know, how long is this really going to be a problem? Because we don't know. We don't know, number one, how long antibody protection will last from the vaccine. We don't know what's going to happen with the new variants as new variants show up. But the plus side of the new vaccine strategies, I will say, is that it's very easy to create new vaccines towards the variants, right? So a company that's making the Moderna or Pfizer vaccine, they can make a new vaccine towards the new variant in a matter of days. That's not the challenge at all. The question is, does the FDA want them to go through the entire approval process again with a large scale clinical trial? Or will they just say, if you show us it's safe enough, maybe we'll let that be good enough? So that's a little bit unknown, but the plus side is that the new vaccine strategies can be adapted really, really quickly to the new variants. Did none of the clinical trials, so to qualify for that clinical trial to be a participant, I believe you couldn't have previous exposure to coronavirus at all. That was an exclusionary criteria for their clinical trial because that would kind of mess up their data. So to enter into a clinical trial, you usually have to answer all these questions about previous exposure, blah, blah, blah. And so nobody in the clinical trial should have been previously exposed to coronavirus. You know, I had heard about the blood type thing, but I think as more and more data was collected, it didn't really amount to anything. I have not heard anything about blood types making a difference long term at all. I remember when that I saw that headline in the news. I never looked at the data. I never saw the data. But the fact that the data didn't kind of make the rounds in the scientific community probably meant that it wasn't particularly strong evidence that there was something going on there. What would have been the best policy from the very beginning to impede the widespread effects of coronavirus compared to the other countries? You know, that's a good question again, and you know, it comes down to, if we look at some other countries where, for example, wearing a face mask is very, very acceptable. And again, if we look at some of the Asian countries where even before coronavirus, people would wear face masks. You know, I think the trade-off there is that some of those countries are also not necessarily places I would want to live in terms of personal freedom, civil liberties, and things like that. So it's always a balance, right? If everyone in this country had to wear a mask from day one, and this was strictly enforced by some sort of, you know, military or police, yeah, we would have lower coronavirus rates. But do we want to live in that sort of, you know, kind of environment? And again, this is a trade-off, and I don't want to get into political commentary about what's right and what's wrong. But I think the idea is that, you know, in some ways, it's not that hard to wear a mask in terms of, you know, it doesn't really take a lot of effort. And if it's clear that it makes a positive impact in the population as a whole, why not do it, right? And yeah, I get the idea about, personal freedoms and whatnot, but I would come back and say, even before the pandemic, remember those old signs that said no shoes, no shirts, no service? Why is this any different, right? It's not necessarily about suppressing people's freedoms and whatnot. It's a public health concern, right? And then that's really what it comes down to. Mm, Okay, there's one hand up. I see Mimi with a hand up. Do you want to, I don't know if you can unmute yourself and you want to ask a question or?
Yes, I want to ask a question. Thank you. I have a question. I got a vaccine last week, my first vaccine, and I did have some symptoms for about a day and a half that were pretty, um, uh, I couldn't do, pretty debilitating. I had a tremendous headache. I was really achy. I had a slight fever, but then after a day and I drank tons of water because they told me at the site that it's a really good thing to hydrate. So I was hydrating and resting and all that. But I thought, whoa, I thought the reaction was after the second one. If I had that reaction, and you said you would, that's actually a good, because you're getting an immune response. Am I likely to get a greater one the second time?
That's a good question. I would say that in some ways, it's, I would say probably not necessarily a lot worse. I would guess that you'll probably get a pretty similar response.
Yeah.
I don't think it would necessarily get a lot worse. And a lot of that again is that soreness and kind of maybe got a little red and it got warm. That's a hallmark of inflammation, right? Kind of imagine if if you burn yourself, you cut yourself, that area gets a little bit red, it gets a little swollen, and it feels really warm. Those are all signs of an inflammation going on. And so when you inject yourself with one of these vaccines, you're also inducing that same kind of inflammation. Um, the inflammation itself is actually part of the innate immune response, the part that I didn't really talk about that much. That innate response is actually there to help the adaptive response. It kind of primes the system. And so the innate response, if you didn't have that response, um, again, a lot of times it helps that adaptive response get better. And so, I mean, I know it's not a very satisfying answer, but it's hard to know. Everyone's going to behave differently. That's why some people have no side effects at all. Other people do feel
Uh,
kind of achy and whatnot because everyone responds slightly differently to these sorts of things.
right. My husband and I got it at the same time. He didn't even have a sore arm. My arm, I could barely lift my arm, and I had a low grade fever too. So I thought, wow, that's really some
Have you
different.
had responses to seasonal flu vaccines before or anything like that?
Just a sore arm.
Yeah, yeah. So it seems fairly consistent for you. And does your husband also get a sore arm from a flu vaccine or not really?
Not really.
Yeah. So it's kind of just your respective immune responses. Yours just is developing more of an inflammatory response, and therefore you feel more redness and more swelling. And that's why, again, everyone's immune system is a little bit different. Even if you
Uh-huh.
take two identical twins, they will have slightly different immune systems.
Uh-huh. Right. Okay. Thank you. Thank
Sure.
you.
We had a couple of questions that came in through email.
Sure.
One was, we read that a significant percentage of healthcare workers are declining the vaccine, saying it was developed too fast. How should we think of their concern?
Yeah, and then that's again a good question. And I think the take home message there is that everyone has a personal level of risk, right? Everyone, that threshold is going to be different for different people. And the best thing I would say is, educate yourself in terms of, look at reliable resources. They joke just because it's on the internet doesn't mean it's true. But, am I personally concerned? No, not really. I think that in this technology has been around. Like I said, it's new for use in humans, but it's been done for a very, very long time in research studies. Um, so it actually surprises me a little bit that many healthcare workers are concerned about it, especially since they've seen the actual consequences of coronavirus infection. But like I said, everyone's going to have different levels of what they consider safe. You know, I know people right now flying on airlines like nothing's different. I know people that don't step outside their front door. And so everyone is always going to have a little bit of difference in what they think is acceptable in terms of risk. And you know, the major risk that has been seen is really just an allergic response. But this is a very immediate anaphylactic response. So I would say in those sorts of situations, that's why when you get the vaccine, they make you stick around for about 15 minutes because they want to make sure you're not having an immediate allergic reaction. That's probably the biggest risk factor right now. But the number of people that have really strong allergic reactions has been really, really small. But I would say that that reaction would be no different than if you're taking any medication, right? You know, when you go to the doctor and you say, oh, I need to take these antibiotics, they're going to ask you, is there any known allergies to those sorts of things? And so, you know, like I said, it's all managing risk, and people will have different levels of that. But me personally, I'm not concerned.
Can we have another question in the chat?
Oh, so this is an interesting question. So, can taking things like zinc, vitamin C help if you are asymptomatic but not feeling sick? So, I was at a conference once, and I was sitting at a table, and we're having lunch, and then there was a photographer at this conference taking pictures, and he sat down next to me, and he's like, "You're an immunologist." You know, I was having this discussion with my wife, and he's like, "She swears by this echinacea stuff, right?" And she says she takes it all the time, and she feels great, and she never gets sick. And he's like, "So does this stuff really work?" And my response is kind of like, if you think it works, and there's no harm in it, why not, right? You know, are there good studies that definitively prove that these things have really good efficacy? Not usually. But that doesn't mean that if you think it works, you know, the placebo effect is a very real effect. You know, when I talked about these clinical trials, one of the things they always do in any good clinical trial is they have a placebo group, because there are decades and decades of studies that psychological effects matter for physiologic responses. And that's why it's critical to always have a placebo group. You know, in a clinical trial setting like this, it's very important because if somebody knew that they had a vaccine, that might affect their behavior. Whereas if they don't know if they got the vaccine or the placebo, then they're not necessarily going to behave any differently, right? And so, you know, the answer to do things like zinc and vitamin C help, you know, there's some evidence out there that it does help, but is that evidence really robust, strong scientific data? Not really. But again, if you think it helps, it doesn't really hurt unless you go really overboard. But, you know, so that's, you know, my kind of sense on those sorts of strategies.
That's good. Another question that came in through email. If an individual has been exposed to COVID-19 and may or may not be positive, should he still get his first vaccination?
That's a good question. And I don't know what the guidance currently is from the counties. I believe that they must have some guidelines on that. I don't know what they are off the top of my head. But I think the exposure, what they might simply do is they might make you wait until you actually are negative, and then you could go in for the vaccine. But don't quote me on that. Oh, I should disclaimer, I'm not a healthcare provider. I'm only here for informational purposes. So I don't know what the county, what the health department is formally saying for that case, but it would be something that you could ask them.
Very good.
I see a hand raised.
Yeah.
Who is that?
Noel, unmute yourself. Noel.
Okay, I think I'm unmuted.
Yes, you're unmuted. Go ahead.
I have a question. I have been avoiding family because I'm over 65 and I'm more at risk than, you know, a 19-year-old or whatever, or a 30-year-old. And when I get my vaccines, because I plan to get them, can I then incorporate my family back into my life? Am I at enough less risk to because they are out there, you know, seeing a lot of people. And so
Yeah.
I've said because of that, I'm going to restrict myself, you know. But is it safe for me to then incorporate them back into my life in a physical sense?
You know, and I would kind of go back to that same argument about everyone's going to have a different level of what they consider safe, right? Because keep in mind that this vaccine is not 100% effective.
Right.
You think that you're going to be in that 5%, you know, is it worth that risk? I would say if you decide to do it, everyone should still be wearing masks. Everyone should still be socially distancing as best as possible.
Right.
Would I feel better about it? Sure. But would I pretend that we're back to normal? No.
Right.
You'd
Okay.
feel much better if all involved were vaccinated.
Right.
So I wouldn't go from nothing to normal. You
Right.
know, could you set up some tables in the backyard and kind of see them again and have a conversation?
Yeah.
Sure. I think that would be okay. But I would, again,
Yeah.
you could treat it like you hadn't had the vaccine. Those sorts of interactions are probably, yeah, okay, but I would still be a little bit cautious, right? Because it is
Right.
not 100% effective.
Right.
But
you.
again, everyone's going to be a little bit different on what their risk assessment is going to be there.
Yeah.
Sherry Woogan has raised her hand. Sherry?
Hi. So since so many of us are in the process of getting vaccines based on our age, if we are with a group of people who all have vaccines, do you recommend wearing masks, or do you think once we're all completely vaccinated, we can?
Yeah, you know, again, that's a good question. So there are a couple things to think about when you look at the data for these clinical trials. Remember, I laid out the system where they had, let's say, 40,000 people get vaccinated. You know, half get placebo, half get the actual vaccine. But their end point was how many got sick. One of the things that is very hard to measure is how many people were asymptomatic. Because if you're asymptomatic, you wouldn't know to get tested. And so if you look at the data about 90%, 95% protection, that is protection from exhibiting symptoms. It doesn't necessarily mean that all these other people didn't get infected, but could have just been asymptomatic. So you do have to be a little bit careful. We don't understand exactly the subtle details between somebody who gets infected and has symptoms, somebody who gets infected but doesn't have symptoms. You know, the question is, if somebody who's infected but doesn't have symptoms, can they still spread it? That is still not 100% clear. And how the vaccine data, again, the vaccine data, a lot of their endpoints were really looking at how many people felt sick enough to go get tested, how many people ended up in the hospital, and then how many people died. Those were their endpoints. They did not really assess how many people actually got infected but remained asymptomatic. So that kind of data is still up in the air. So I would still be very careful, even if everyone you knew had been vaccinated. There's no guarantee that there might be somebody in there that might have the virus but just feels perfectly fine. And you don't want that to begin to spread. So, you know, again, it's a personal safety, what level you're comfortable with, but I wouldn't assume that just because everyone has been vaccinated, everyone is completely safe. That is an assumption that hasn't formally been tested yet.
I think we have one final question in the chat.
Can the vaccines make you sterile? As far as I know, there's not been any evidence that that is a worry. You know, if you think about it conceptually, there's nothing in there that would be a concern. You're getting a shot in the arm, you're getting an immune response in the arm. The immune response there should not have any implications on, you know, reproductive health in any way that is, you know, foreseeable in any way. So I don't think that has been a concern for these vaccine developments.
Okay, well, with that, I think we've asked all our questions. Professor Lin, thank you so much for this very informative presentation. We really appreciate it. You've made us a lot smarter today, hopefully going forward. Absolutely, really appreciate it. Again, thank you very much. You brought it down to a place where it was easy for us to understand, and you responded to our questions. So again, I thank Mimi and for bringing our presenters to us, and Mary Costa for backing me up. It takes a village. So thank you all. Next month, James Masters regarding the census. Thank you all. Have a great day.
Thank you, everybody.